what is an alveolus?
You look on the table, it varies depending with exercise, but it drops from 21 to about 14, about a third. If you exercise, if you make more CO2, then that's going to drop even further. Okay. All right, so using Dalton's law, of atmospheric pressure at sea level is 760 millimeters, and the oxygen molecule from approximately 20.9% at an elevation of 12,000 feet, which is 500 millimeters of mercury. What proportion of the air is oxygen? A is less than, B is more than, and C is equal to 21%. And while you're working on that, I'm going to do some math. Oh, why I have to do this? Looking for a calculator, it should be under accessories. There's no accessories. Okay, so at this atmospheric pressure of 500 millimeters of mercury, instead of our PO2 being 160, our PO2 is going to be 105. Right? So it's going to be about 65 inside our alveoli instead of 104. Okay, because this is atmospheric. So we drop our atmospheric pressure from 760 to 500. Then it's still, what was the answer here? C. So it's still 21%. So to find out what the atmospheric partial pressure of oxygen is, you take 500 times 0.21. And that gives us an atmospheric pressure of oxygen at 105. So it drops from 160 to 105 when we go from sea level up to atmospheric pressure. That's atmospheric. An alveolar would be even less than that. Because when atmospheric oxygen had a PO2 of 150, if you take 14%, I'm assuming, I mean, this is just the medical number, but it should be 0.14 of 760. Let's see what we get if we do that. And that, again, that's going to be variable. That's going to be variable depending on how fast you're breathing, because you'll be hyperventilate, you're going to breathe off CO2. And if you're exercising a lot, you're going to be making more CO2. All right, so that's going to, and you don't change your respiration. All right? All right, so then the solubility law, which is Henry's, Dalton's was the proportion of a gas, pressure of a gas is equal to its proportion. So Henry's law says that a constant temperature, the amount of a given type and volume is directly proportional to the partial pressure of that gas. So that means that in the blood, our gas is 21%, oxygen is 21% of the gas pressures in the blood. Okay? Well, not 21%, but equal the 14% we saw in the alveoli. So the more pressure we get, this is why if you're a scuba diver and you're breathing air under pressure, and most people breathe mixed air in the tank, they don't breathe pure oxygen. So if the air in your tank is just under pressure, it's still going to be 21%. Okay, and you're going to have it. But you're going to have a lot of gases dissolved, including nitrogen, dissolved in your blood because you're breathing it under pressure because you're surrounded by. Does anybody know how many feet deep in the lake or river or ocean 1 atmosphere is equal to? 33 feet. So if you are 33 feet underwater, that's 1 atmospheric pressure. Okay? So within that, you can move up and down and not worry about how much time you're spending under the water. Then once you go past 1 atmosphere, you have to start calculating the number of minutes that you're breathing the pressurized air. Okay? And then give yourself time for your lungs to equilibrate as you move up. because your whole body is under pressure, including the air that you're breathing. So why don't we talk about temperature? This is showing the solubility. The carbon dioxide is more soluble than oxygen, which is more soluble than nitrogen in the fluid, okay? And so when we have, notice which way the solubility is moving with temperature. As the temperature increases in the water, does solubility go up or does solubility go down? It goes down, which makes sense when you think about adding heat to a pot of water. You're increasing the energy and the movement of the gas as well as the water molecules, and they start to come out of solution, okay? So, anybody getting married this fall or winter? All right, so there's these little cool things you can do for showers or wedding, and that's to freeze balloons, edible balloons in the ice. Now, if you just take it right out of the tap water, congratulations, by the way. You just take it right out of the tap water and you freeze it, the ice is cloudy. All right, if you just turn on the faucet because of all the air that's been mixed with it. So what do you do if you want clear ice? Boil it. Or freeze it under vacuum, and none of us have vacuum freezers that I know of in our home refrigerator. So you boil it, okay? Now, you've got to cool it, and as it cools, it's going to pick up oxygen as well. So you've got to figure out how to boil it and then add the flowers to it without cooking the flowers. So I never actually tried it, but so let's see if we're ready for our third question. We'll come back to this one. Which of the following has the greatest amount of living material? The South China Sea, which is where I grew up, or the Caribbean, that's what surrounds the Philippines, or the Caribbean Sea, or the Arctic Sea? What did we just say about the amount of oxygen? So, the warmer the water gets, the less oxygen is dissolved in the water. What does living mass need? Plants or animals? Oxygen. So it might be more colorful in the tropics, but when we look at total biomass, like the tiny little plankton, okay, the colder water is going to have the higher amount of oxygen. That's how huge whales can live up in the Arctic, because of the amount of total living mass that's up there. Because there's actually more oxygen available. Plus, you don't need as much. You don't need as much oxygen, right? Okay. So.
No, that answer is solubility. You go back to the main
Yeah.
definition
Yeah.
about.
Yeah. So solubility is how much. We're not making a chemical reaction here. What we're doing is taking a gas, and we're sticking with oxygen right now, but it could be carbon dioxide. We're taking a gas and asking it to move around in between water molecules and not rise to the surface and come out of the water. That's solubility. It's soluble in the water, just like you would take sugar and dissolve it in the water, or salt. It's not going to precipitate and sink to the bottom. If it was plastic beads, it would never be soluble in the water, okay? So this is soluble in the water. It makes weak chemical interactions probably because of the polar nature of the water. Oil's not soluble in water because of its non-polar nature, but it mixes equally and stays in a mixture in between the water molecules. So we get more, if we go back to this prior image here, because the gas has an energy, it's going to want to come from move out to where there's more space to move around, all right? But if we put higher pressure on it, we get more gas staying in amongst the water molecules. So as we add heat to it, we're adding energy from this end, and it's going to move up out of the water into the air, okay? So when we talk about PO2 or PCO2, we're talking about this right here. The oxygen or the carbon dioxide is a gas in and amongst the water molecules. It's not bound to hemoglobin. It's not forming carbonic acid or bicarbonate ions. It's the gas form, and it's in the water. Okay. So if you rise too fast after breathing air under pressure in a scuba tank, then that gas comes out of solution in your joints. It forms bubbles. It can form gas air bubbles in your tissues because it's coming out of solution faster than you can breathe it out of your lungs and have the bubbles rising. Okay? So this is just, I'm not going to test you on this. This is just an interesting comment about a fish that doesn't have any hemoglobin. We would not be able to live without being able to transport the oxygen bound to hemoglobin. We don't have enough oxygen dissolved in our plasma to provide oxygen for the energy that we need to make in our mitochondria. Okay? So we have to have the hemoglobin available for the additional transport. That would not that doesn't affect PO2. It's dependent on it, but it's not measured by PO2. We measure it by hemoglobin saturation, how much oxygen is actually bound to hemoglobin. So in these fish that don't have any hemoglobin, all right, they don't have a very high metabolic rate. They're not needing a lot of ATP. Plus, they have some changes in their cardiovascular system and circulatory processes because they only have the oxygen that is dissolved in their tissues and they live in cold water. So it has more oxygen available to move across their gills. All right, so looking at the next thing, if we go back to our previous slide, we had partial pressure and solubility, okay? Then, how much and how long it takes for the gas to move across also has to do with distance. If you remember from Bio 430, we looked at factors that affect diffusion. We had distance, how far does it have to travel? Because the farther it travels, the farther apart it gets spread. So if it has to travel really far, it's getting less and less concentration, and that slows the probability that it might bump into another molecule. All right, so diffusion thickness, the respiratory membrane thickness is going to affect this. And this doesn't typically change unless there's a disease process. So pulmonary edema, when we talked about pulmonary edema last week, was that an obstructive disease or a restrictive pulmonary disease? So the narrowed airway with a large alveolus was obstructive. A normal airway with half the alveolus blocked off was restrictive. So pulmonary edema is going to be restricted. We lose volume. We don't have a narrowed airway. We lose volume available for gas exchange. Okay? So pulmonary edema up here where we have fluid in the lungs. Pneumonia would be the same thing. Or pulmonary fibrosis where we have additional amounts of connective tissue, additional collagen fibers. So in the first one, if we're talking about pulmonary edema or pneumonia, where we have a fluid, that's going to increase the diffusion distance. Because it has to diffuse through that fluid as well as its normal distance to get to the blood. And that's going to increase the time it takes to move from the alveoli to the blood, or if it's carbon dioxide, to move from the blood to the alveoli. Okay? In addition, if we add extra collagen fibers, that can create a barrier to the diffusion of the gases, and we can actually increase the distance. So not just make it more solid material with the protein fibers, but actually spread apart the two simple squamous epithelia and increase the distance. So, based on rules of diffusion, what do we do to treat this? Not to treat it to get rid of it, but to provide an adequate oxygenation. So if someone has pneumonia or pulmonary edema or pulmonary fibrosis, how can we overcome the effect of diffusion to increase the amount of oxygen in their blood or increase the rate at which CO2, because otherwise they're going to build up CO2. They're going to become acidic because CO2 is not leaving at fast enough a rate. So we need to increase the amount of oxygen moving in. That's a little easier to do than increasing the amount of CO2 moving out. What can we do to increase the amount of oxygen moving in? Give them more oxygen, all right? So a cannula with, instead of 20%, maybe 30%, all right, or a higher pressure. Is pushing air into their lungs to increase the CO2 here, all right? We can't create we can't do anything about this physically. We can certainly help to get rid of the fluid, but as far as CO2 moving, the best that we can do is to decrease the CO2 on this end by better ventilation, okay? So deeper breaths, getting them to breathe more deeply and more often to get this air out and keep the PCO2 in the alveoli lower. That's going to increase the gradient from Thursday. Remember we talked about two components that affected the pulmonary ventilation. We talked about several, but one was compliance and one was elasticity, and they were counterpoints to each other, all right? So when we had that ventilation tube attached to the sheet plug, and we were pushing air in, the fact that there was no surface tension helping to keep the sheet lung expanded, did that increase, or, so, well, first of all, what's compliance? The ease of expansion, all right, so opening up, breaking the surface tension of those alveoli, and moving air into the alveoli. So if air moves into the alveoli easily and the lung expands easily, is that high compliance or low compliance? High compliance. So that if it expands easily, it has high compliance. If it doesn't expand easily, if it's a stiff expansion, then it's low compliance. The recoil is due to elasticity, okay? All right, so pulmonary fibrosis would provide low compliance, would make it difficult to expand the lungs, okay? Pulmonary edema, is that a form of obstructive pulmonary disease?
No.
What was the other form? Restricted, all right? So again, the terms can be confusing because you would think if you obstruct airflow, then you've lost. So think of obstructive again as a narrowing of the airway, all right? And a loss of alveolar walls, like an emphysema. Restrictive is a loss of volume available for the gas to move into. So with pulmonary edema here, we have taken up part of our volume. We've decreased the amount of air that can move into the alveoli because it's got fluid taking up 10% or 20% or 60% of the space. So we've lost the ability to move air into it. And then A is both, and C is both A and B. There's just one correct answer, because I gave you the combination as an answer choice. Okay, ready to move on? Okay, surface area for gas exchange. This is almost common sense, because we need surface area, right? So if we lose surface area, it's gonna take longer for the gases to move either for or across, all right? So this can occur with both restrictive and obstructive pulmonary diseases. So obviously, we've lost surface area here with the pulmonary edema, okay? Because it's not been completely lost, but it's gonna take a longer time for the distance. How do we lose surface area with emphysema? Anybody remember? Remember the drawing I drew of emphysema? So with normal alveoli, we have the alveolar walls, that's our surface area. And with emphysema, there's a destruction or a loss of those alveolar walls. That's what makes it easy to expand the lungs, because we have decreased surface tension. We have fewer surfaces to have a surface tension. So there's a loss of alveolar walls, all right? And so there's a loss of surface area. So that can occur not so much with asthma, all right? Asthma is narrowed because of the mucus in the airways and the constricted respiratory tube, but the alveoli aren't really affected, okay, unless the mucus gets way down deep. Mucus is typically described as being within the bronchioles. So that's more of a narrow airway. The emphysema affects the alveoli, and then the airway narrows in emphysema, but it narrows when you're trying to breathe out. Remember we talked about how you're trying to force air out and you squeeze your expiratory muscles, and it's that trying to force air out that actually narrows the airways. It's not the disease process as much as it is in asthma. All right, so just showing edema and how we're losing volume and surface area with pulmonary edema. Just more of the same. You can read those on your own. All right, so alveolar ventilation perfusion coupling. Perfusion has to do with the heart, all right? So what we're talking about here is a coupling of blood flow with gas exchange. So let me come down here and get one of our alveolar models. Actually, air sac models, both. The blood in the pulmonary capillary is not going to flow by one alveolus like I have drawn up on the board and completely equilibrate so that we reach 104 millimeters of mercury, all right? So as the blood is flowing by here, and oxygen is moving in, it's not going to instantly become 104. It has to flow by one alveolus, and then another one, and then another one, as you move gas into and out of your lungs. And by the time it leaves, it has equilibrated, okay? So it's going to be going by thousands of different alveoli, exchanging carbon dioxide and picking up oxygen as it flows by those different alveoli as you breathe air in and out. So it's not just one pass by one alveoli that brings us to that equilibration. Now, if you go into respiratory therapy or work in a pulmonary unit, you're going to get into all of these tests and pulmonary ventilation components a whole lot more. But in the organ, if you are sending oxygen to a skeletal muscle tissue, all right, and you're exercising particularly your lower limb muscles instead of your upper limb, although we usually pump our arms, but it's your lower limb muscles that need oxygen as they use up the oxygen in the myoglobin and make ATP and all that kind of stuff. They need more oxygen to make more ATP. You're going to be doing aerobic exercise. So what happens to blood vessels that are carrying blood flow to tissues that show a drop in their oxygen levels? The arterioles are going to dilate, so more blood flow goes to that particular tissue, all right? The smooth muscle, remember the nitric oxide we talked about with the penile arterioles? Same thing in tissues that need additional blood flow. The lining of those arterioles sense the low oxygen around them, and they release nitric oxide, and the smooth muscles relax, and more blood flow goes to that tissue. So let's say we have an area in the lungs that we have pneumonia, okay? So we're not getting much oxygenation across that respiratory membrane. And as the tissue, as the alveolar capillary blood goes by, it's not getting much oxygen. That's a waste of blood flow. If we were in our systemic system, and this had low oxygen, all right, available, the blood flow to this supply would increase. Because the purpose of our systemic capillaries is to provide oxygen to the tissues. The purpose of our pulmonary capillaries is the reverse. The purpose of the pulmonary capillaries is to receive oxygen. So if they're not getting much oxygen from these particular alveoli, there's no point in wasting circulatory time to go by them. So the arterioles constrict and cause the blood flow to be diverted to other areas of the lung that are receiving adequate oxygenation, all right? So when we have systemic tissues that are low in oxygen, systemic arterioles dilate. When we have lung tissues that are low in oxygen, pulmonary arterioles constrict, because there's no point in sending blood to an area that doesn't have adequate oxygen for it. Okay? So the whole purpose is to redirect the blood flow to areas of the lung that have oxygen available to them. And that's the general why we call this a ventilation perfusion. It's the blood flow coupled to the oxygenation ratio that's available to the tissues. Okay, any questions on the previous activity? So it's thinking about diffusion. What are the factors that are important to diffusion? Concentration gradient, distance, all right, are the primary two that we see affecting external respiration and the movement of both oxygen and carbon dioxide across. So I actually put all these little O's in there before I figured out how, a long time ago, before I figured out how to group, where do I want? Arrange, where you can group everything and then just copy that and put them in different locations. So a lot of copying and pasting individual O's. So one of the oxygen carried 3. Oxygen is carried by 2. 1 is dissolved in the plasma, what we just got through talking about, affected by the solubility of the gas and the temperature of the gas. So that's represented, I'm going to erase my pulmonary fibrosis here and redraw it without such a thick area. So, get rid of the edema. So the oxygen that we see here, the blue circles, all right, the white circles over there, represents a small amount of oxygen transported by the cardiovascular system. It is that oxygen that is contributing to our PO2, okay? So if we have a high PO2, that means we have a lot of oxygen dissolved in the blood. If we have a low PO2, then this number is less, okay? Same for PCO2, it's one of the ways, for carbon dioxide we have 3. As I said earlier, we cannot live on only the oxygen dissolved in the blood. That's why you feel tired if your hemoglobin is low, okay, because it's not providing enough oxygen transport. So here we have oxygen bound to hemoglobin. This is binding to the ion associated with the hemoglobin molecule. So remember we had 2 different types of hemoglobin, and each hemoglobin molecule, and each hemoglobin molecule had 2 types of proteins, globular. And iron, we had 4 ions, each one capable of binding an oxygen gas, O2. So what we actually have is a combination of this, all right? Because, why does this not exist? Well, it can to some extent. The only time you would see something similar to this is heavy carbon monoxide poisoning that had passed through the tissues, and that dissolved oxygen had moved into the tissues, but because carbon monoxide was bound to the hemoglobin as well, oxygen cannot let go. It's not going to be 100% like this, but that's about the closest situation to where you would see a low PO2 with saturated hemoglobin, okay, and that's due to the effect of carbon monoxide. So 2 different ways of transporting. Dissolved in the blood, that's our PO2, and bound to hemoglobin, and we call that hemoglobin saturation. Okay. You may have heard about somebody's SAT levels. So when my daughter-in-law went in for her gallbladder surgery last February, her SATs were at 50. Okay. Typically, they won't send someone out of surgery after recovery to the floor if their SATs are below 95. Okay. So they wouldn't even do start surgery on her with her SATs at 50 in the 50s. So this is usually what we see. Oxygen dissolved in the blood and bound to hemoglobin. Okay? So here's our hemoglobin molecule. As I said, we have 2 different types, alpha and beta. We have a 3rd type, which is the fetal gamma hemoglobin. And I'm not going to get too much into the chemistry of this, so some of these slides are a little bit hairy. So listen carefully, because I'll tell you what you need to know and what you don't need to know, okay? So in the hemoglobin, we have this, what's called a porphyrin molecule, and it has the iron in the middle of it. And it changes shape, depending on whether oxygen is attached to the iron or not. Why is there such an attraction? I'm leaving out the word strong and weak. Why is there such an attraction between iron and oxygen? Remember what I said about the chemistry of oxygen? It has a need for 2 electrons to stabilize its electrons surrounding, and iron has an electron it can give off, has electrons it can give off each. We usually see it like Fe plus 2 or plus 3, because there's a variable, all right? So this plane, you don't have to learn all this chemistry here, but the porphyrin and the iron I want you to know, and that flexes, depending on whether or not it has oxygen or not. It will flex and change shape. So here, it's in a kind of a tinted form, the porphyrin molecule, when oxygen is not attached, and when oxygen is attached, it flattens out. Okay? Yeah. The histidine has to do up there with sulfate bonds, and we're not going to get into that part of it. Okay? But I just want you to know that when oxygen binds to it, it changes shape. because that's going to affect affinity and how easily the oxygen stays on or doesn't stay on. Affinity is a chemical attraction with electrons being shared or exchanged. So this is just a little video animation showing the changing in the planar form of that porphyrin molecule. All right. So the four boxes up at the top represent the, on each hemoglobin molecule, there's 4 irons. So it represents each hemoglobin molecule can bind to 4 oxygen. And if you've forgotten your chemistry, K stands for affinity. Okay, the strength of the chemical attraction between the electrons forming the bond. So when the first oxygen binds, it makes it easier for the second oxygen to bind, and that makes it easier for the third, and that makes it easier for the fourth. In reverse, that fourth oxygen doesn't come off very easily, okay? And it's less easy for the oxygen to come off as we move in reverse. So I want you don't need to know these shapes, but I want you to know the difference between deoxy and oxy. It's kind of self-explanatory. So deoxyhemoglobin is when the iron doesn't have oxygen attached to it, and oxyhemoglobin is when it does. Okay? So when you use those terms and we go back, it's similar to oxygenated blood. So this would be deoxyhemoglobin here, and this would be oxyhemoglobin, if we're looking at it in the sense of the protein itself. So when the blood is 100% saturated, all of the hemoglobin molecules are transporting 4 oxygen molecules. When the blood is 50% saturated, each hemoglobin is carrying 2. It's not that 50% are carrying 100 and 50% have nothing. So when oxygen is 50% saturated, the hemoglobin, each hemoglobin, this would be 50%. Each of the hemoglobin molecules would look like this, okay? Or 75%. In other words, when we say that your hemoglobin is 75%, we're saying 75% of each hemoglobin, 3 of each of the 4 sites, has oxygen bound to it, rather than 75% of the red blood cells have oxygen and 25% do not. So it's going to be distributed. We're going to put oxygen on every hemoglobin and then add the next one, okay? And a dispersion of oxygen components. So we're going to take a break here. It's a little early, but this is an important illustration of the relationship between hemoglobin saturation and CO2 of oxygen. I want you to have fresh brains for that. So when we come back, we'll start with this slide.